Today about using data and its impact on health. And it's actually going to be fairly broad. I'm going to talk because it's all the program, so I thought I'd have a lot of health-related information. And then the last part, in fact, we'll zoom in on the Exposome. Let's see, do you use?
Keyboard, right or left or down?
No, I was thinking more for the mouse to see. Let's see. That works. But then I'll stand. Let me move this a little closer then. So I can use the mouse so people from afar can see. Great. All right, so this basically reflects what Gary just told you. That is the way we think about health. It's the input of many things. It's the input of your genome, your DNA, then all the various things you're exposed to, the so-called exposome. And I'm a believer that in the not-so-distant future, people will be born with their genome sequenced, and then one will want to know what kinds of exposures will lead to what kinds of health outcomes in some probabilistic sense. That is to say, if you have a mutation that predisposes you for Parkinson's, you may not want to be a pesticide worker. Now, you may not want to be a pesticide worker anyway, but you especially don't want to be one if you're predisposed for Parkinson's genetically. And that's the sort of thing we'd like to understand, again, in probabilistic terms. So I think as many folks are aware of, we're now in an amazing time where there's been a revolution in all kinds of technologies. This is really just a subset of what's out there. That is to say, genome sequencing and sequencing in general. Now lets us sequence genomes pretty inexpensively. We pay $150 for a human genome sequence. That will drop quite a bit next year. Not the interpretation, just the sequencing. Many of you appreciate there's been a revolution in mass spectrometry, so you can look at bodily fluids like urine and plasma and profile thousands of features, maybe about 1,000 of which you know what they are, but nonetheless, it's quite a few. And I will argue there's been a revolution going on in the wearables technology as well, so that you can now measure many things, and I'll spend a fair amount of time on that as well. So we're now in a situation where you can measure all this, and what I haven't put up there is there are now a lot of people surfing through people's emails and the way people look through websites to actually figure out behavioral patterns, another form of data that you can collect and try and understand people's psychological states based on the way they interface with their computer. So many of you may know, a number of years ago, we actually set up something we call personal omics profiling, where the idea is that in addition to getting your genome sequence, we'd make many other measurements as well. In fact, these days we follow 14 different ohms on folks of all sorts of type you'll see here. I'll get into some of this a little more later. And then about 4 years ago, maybe 4.5 now, we added on a lot of wearables information, and I'll present that in quite a bit of detail as well. And the general idea is that what we do is we sample people roughly every 3 months or so when they're healthy, but when some sort of perturbation comes along, like a viral infection or maybe colonoscopies, what have you, we'll take many more samples to try and understand what's going on. Now, why are we doing all this? Well, we're really trying to define people's personal baseline health state. These days, you may know, medicine focuses very, very heavily on disease states. We tend to profile people when they're sick, but we actually don't do much to study them when they're healthy. And we don't really know what a healthy profile looks like, how does it change over time, how does it differ between different people. So we're spending a lot of time trying to just define in incredible detail what it means to be healthy, and then what happens when people do transition to disease. We're also trying to understand how the different ohms relate to one another, because I'm a believer if you know all the inputs into a system, you should be able to predict outputs. As an example, if you know someone's genome, epigenome, immunome, and the food they eat, probably their microbiome, you should be able to predict their metabolome, I would argue. We can't do that now, but that should be possible in the future. There's modest amounts of that going predictive ability now. We also want to know how individual responses are both similar and different from one another. And probably from a health perspective, this last point is really important. As we profile very deeply, we may learn things that impact their health, and that might be useful for managing their health. And it turns out that happens quite a bit, especially when people get older. You do learn things that will be important for their health, and you'll see some of that. So many of you may know we actually started this project now in March. It'll be 8 years. So we've been doing this for quite some time. We started just to get the technology going. We actually started with me. And I didn't know that I would be interesting, but it turns out everybody we profile is interesting in lots of different ways. So it turns out that the general scheme is shown here. Again, we profile people every few months when they're healthy, and then when they go through some sort of viral infection, and over this time I've been through 11 viral infections, including one just fairly recently, we'll take many more samples. So 9 months into the study, I had a respiratory syncytial virus infection, and so these are day numbers. Basically, we take blood as soon as I get congested, then next day, this case, 2 days later, let's see if I can get that back, 2 days. So first day, next day, 2 days later, 2 days, 3, so on. So we do dense profiling again when someone gets ill, and then again, 2 to 3 months, or at other interesting times as well. I forgot to say, but I've also had Lyme disease, had a bike accident about 2 summers ago. I don't look for these things, but if they happen, I might as well get good data out of it. That's how I view it. All right. So anyway, again, I'll go through this kind of quickly. We published because I want to get to the newer stuff. But basically, we started with my genome, which made many predictions, a lot of which match what I knew about my family history already. So I have high cholesterol. I knew that. My genome says that as well. And low risk for obesity. Nobody in my family's ever been overweight, this sort of thing. One big surprise was that I was predicted to be at high risk for type 2 diabetes. So I have many risk alleles for type 2 diabetes, and when you sum them up, I'm actually at the far end of the distribution virtually compared to everyone else we've sequenced. And it turns out that sure enough, because we're doing all this dense profiling, in fact, my glucose, which had been going along perfectly normally, did shoot through the roof. In fact, to the point where I was classified as diabetic. Okay, we only discovered this because I was actually starting to look at insulin resistance and things like this because of my genome. And as we were following my fasting glucose pretty carefully, as I say, it did go up quite high to the point where it's classified as diabetic. That's this time point here. That's April 11, 2011. I know that because up until that point, I had the world's worst diet. I ate all kinds of sweets. I would eat those cookies out there. I've cut all that out. I doubled my biking, started running, and don't think this is a short period. That's 8 months to get that back down to normal. So this made a big splash at the time because the first time someone used their genome to predict risk for a disease, and in my case got it, and I was able to manage it. It's also quite interesting because when it occurred right after this nasty respiratory syncytial virus infection. So the hypothesis is that I'm genetically predisposed for type 2 diabetes, and then in conjunction with this pathogen, which is common in kids but not as common in adults. But the idea is that when I got exposed to this, it actually triggered the disease. That's the hypothesis. We're sort of testing that on a larger scale. I can tell you anecdotally, there's been lots of other folks who've written to me who have had similar situations where they become type 2 diabetic. I'm type 2. It's well known, by the way, type 1 diabetes is associated with viral pathogens, although exactly which ones is still unclear. But type 2 has not been so clear. Stresses are known to do that. Anyway, one criticism out of all this work, and justifiably, was that, well, Mike, it might have gone away on its own. So we accidentally did the right experiment. You'll see as we scaled to study 105 people. Basically, that's a big project. You'll see in a minute. We're collecting all these samples. Nobody was looking at the data. So I'd stopped running, and then right about here, somebody looked at the data and said, Mike, you realize your hemoglobin A1C, that's a better measure for steady-state glucose. You're actually 7.0. I didn't say, but the threshold for being classified as diabetic, 6.5. So I did cross that threshold the first time. And then it turns out when someone looked at the data, sure enough, I was classified as diabetic. I was 7.0, so they said back to diabetic. Went back and looked at data. Sure enough, it was running quite high. And I had stopped running, so I started running. I got it down to 5.7. That's about 2 years ago, and it's been creeping up ever since. It was in the low 6, actually more like 2.5 years ago. Kept creeping up to about 6.4, and you can't tell, but I actually switched from running to weightlifting. You can say you can't tell, I'm sure, but the point out of all this is that the idea is muscle mass is better for glucose homeostasis. Turns out that failed miserably. I kept going up, and so I'm back to diabetic. I'm 6'8", And the point is I'm now actually adjusting my diet to try and do this, and I'm about to go on metformin. So anyway, the point out of all this is that there's no question I have a glucose misregulation problem. Turns out I have 2 brothers, 3 sisters. They came out to visit. We took blood from everybody. And sure enough, they're all elevated for glucose too, and they're as thinner, if not thinner than me. So there's no question we have this running in our family, and it was all triggered by the genome sequence to see this. Okay. So anyway, so we would argue genome sequencing is valuable for finding these sorts of things. So the other thing that we've looked at, we've now extended the study in many ways. We've looked at microbiome, which is pretty cool. I won't tell you about that. Here's what we've done with the epigenome, which we would argue is very, very important. And obviously, when you think about exposome, the epigenome must be impacting you. The exposome must be impacting the epigenome in ways that nobody's really looked at, right? What does a viral infection do to your methylation or chromatin? It's not so clear. So, but to back up, so I think all of you probably appreciate that your DNA methylome, if you will, it's one form of epigenetics. It's heavily influenced by nutrition, by lifestyle like exercise. In fact, one of my favorite studies is one from Sweden where they had 23 people. You may know the study. They biked with one leg but not the other for 3 months, 4 times a week. And they took muscle biopsies before and after biking, and the biking leg and the non-biking leg. And there were 4,000 methylation differences in the biking leg relative to the non-biking leg. So that's a big deal because what it says is that exercise chemically modifies your DNA. And we know that, not necessarily directly, but, well, there are interesting pathways that probably affected. But it does it in a way that presumably leads to changes in gene expression because it's well known that DNA methylation does influence your expression. So methylation promoters is associated with gene silencing. So that's kind of cool. So we've looked at the first 3.5 years of my time course where I've been through 6 viral infections. And by RNA analysis, I can tell you that my RNA bounces around all over the place. Not surprisingly, every time I get 6, my immune response goes off. And you can see it's a very strong signature. I didn't show that. That's pretty obvious. There's some other interesting patterns too, like neurological markers that change and things. Turns out the methylome only changed twice. This is out of my peripheral blood monocyte cells. It only changed twice over these 3.5 period. And those 2 times are actually the 2 times when my glucose went off that I showed you before. So that, going back to this other diagram, so it went off, here and here. It's probably hard to see, but it shows up here. So the 2 times when my DNA methylation changed, it's fairly subtle, but it's definitely very clear. It turns out it's the time when my glucose misregulated. And on top of that, it turns out it slightly precedes when my glucose actually hit its peak times. So we think it's an early marker, DNA methylation, for chronic disease. And given that methylation is affected by aging and other things, it's pretty reasonable to think that in general, DNA methylation is a marker for chronic disease, as opposed to transcriptomes, which bounce around quite a bit. So anyway, and I forgot to say, but the genes that are closest to the methylation changes turn out to be involved in glucose control. So the point out of all this is that we think methylation is having an effect on metabolic function, which in turn is affecting my glucose levels. So since that first set of studies, we've actually now expanded this, as I mentioned, to study 105 people. We're now doing everything to them that we're doing to me. So we sample them every 3 months when they're healthy. And when they go through some sort of perturbation, like a viral infection or a stress response, a colonoscopy or sleep deprived, what have you. A number of them are doing dietary studies. We have that running as well. We actually sample them much more frequently. And so we're trying to see the effects of viral infections, colonoscopies, et cetera, antibiotic takes on the profiles of these folks. It turns out there's a ton of data here. Looks like a slide got left out. What's powerful about all this is we've now collected about 1,800 time points We've analyzed the first 1,044, and we've done these deep omic profiles on those, and all that information is going to be publicly available. Most of our folks have agreed to make their data, including their genome sequences, public, not their names and birthdays and such, but you can actually get access to this information so you yourself can explore it. So if you have a cool project, you can dig in there and look through the data yourself. There's a few that have their data restricted, but the vast majority are making their data completely open access. Okay, so just to give you a few vignettes, one thing, criticism that comes up is people say, Mike, you learned something about your genome sequence, but you're probably an anomaly. That is, most people may not learn much from their sequence, and maybe it's the opposite. Maybe what you're going to do, in fact, a lot of people say, is what you're doing is actually wrong. You're actually sequencing people's DNA and turning everybody into hypochondriacs. Okay, that's one concern that is out there. And so I always find that a little alarming because people are very comfortable with family history. That doesn't necessarily turn people into hypochondriacs, but genetics does scare people. So anyway, we've now, for at least 70 people, we poured over their genomes like mine. 12 of them have clinically actionable information. That is to say, they have a mutation in their genome that leads to a test when they go to a doctor's office to look out for something, much like you might do now if you know heart disease or something's running in your family. And just to give you a few examples, here's a person with a BRCA mutation. This happens to be a male, but he's at increased risk for breast and prostate cancer in his case. So he actually gets screened for that more frequently than he would otherwise. And there's a slew of other things as well. About half of these genes are on something called the American College of Medical Genetics list. Those are the ones in red. The other half are actually genes. We did a very comprehensive analysis of the genome. So we find mutations in other things that is, again, important for people to know. Like, for example, some of these, this one here, it turns out this is a mutation that leads to potential cardiomyopathy, and it turns out this individual, who's fairly young, was discovered to have a heart defect on a stress echo. So in fact, this turned out to be useful. He's on medication now as a consequence of us sequencing his genome. And there's another person, this is an interesting one, this is a plug for precision medicine, but basically, one of our folks, I forgot to say, but about half our folks are pre-diabetic, and there's a few of them that are diabetic. This individual was classified as type 2 diabetic, but it turns out he has a different kind of mutation. It's called a MODY mutation. It's more Mendelian, and the way you treat a MODY person is 100% different from the way you treat a type 2 diabetic. Okay, it's a whole different medication. In fact, there's a famous story of a type 1 diabetic who late in life got his genome sequence, and turns out he was a MODY. He estimated he had 21,000 insulin injections over his lifetime, and had they discovered this early, he probably would have needed a total of 0 because he would have been on a different medication and not have actually watched his insulin go up and blow out, et cetera. So the point out of all this is actually getting the right information can be extremely valuable. And so we would argue turns out 2 to 4% of type 1 and type 2s are MODYs, and nobody has no idea. Nobody screens them. We think you could do it for $50, by the way. All right, other things we found. Well, we found in addition to one heart defect, we found another person with a heart defect. We caught someone with early lymphoma, someone with elevated IgM cells. It's like a pre-cancer. We found quite a few people who are, turns out, pre-diabetic who had no idea, some of them fairly elevated in their glucose. A lot of you, in fact, I guarantee there's a lot of you who are spiking your glucose and you have no idea of it. That is to say, you don't have strong glucose control, and you will probably become diabetic. 9 out of 10 pre-diabetics have no idea they're pre-diabetic. Okay, and that's probably a number of you, not to scare you. Okay, I guess I did, sorry. But anyway, go get your glucose checked. All right, this slide got out of order. This is the one I was saying before about things being open access. I think I'll skip over this other than to summarize that we actually have a lot of people doing these dietary studies. This is a particular one where people, maybe I'll summarize quickly. They basically, 23 people went through a weight gain, weight loss cycle where they overeat 1,000 calories a day for 30 days, gain 6.5 pounds on average, and then they lose weight for the next 60 days, come back to baseline. We sample them before, after they gain weight, after they lose weight. And I can tell you half of them are insulin resistant, which is a It's like an early stage of type 2 diabetes, if you will. And then the other half are healthy controls, BMI matched. And I can tell you, no matter which ohm we use, we can distinguish the insulin resistant from the insulin sensitive folks, these healthy ones. This is just a metabolomics profile. You can find markers that, again, each row is a marker, each column is a person. These are markers that associate with the resistance, these associate with sensitivity. Some of them, like this one, comes from the microbiome. So you can actually, so we now have a predictive test for insulin resistance that we think can be done as a metabolomics test that probably would cost about $50 as well, whereas the current method for doing this is something called SSPG assay. Takes 6 hours, costs thousands of dollars. And so we think these simple markers can be a much, much better way, and probably will actually stratify people slightly differently as well, we think, for other reasons. I'll just tell you that when you look at people who gain weight, you can see all kinds of pathways changing. Inflammation goes up. It turns out this one, the hypertrophic cardiomyopathy goes up pathway. We think people who gain weight are well known to get heart attacks. So we think this is a molecular explanation for what's going on. The other thing I'll tell you is that at baseline, everybody's different. So we all have very different profiles. In fact, it turns out longitudinally, people don't change much. I'll show you one slide on that in a minute. But it turns out if you compare people with one another, we're all very, very different. It doesn't matter which home we use. This is just the cytokine profiles. This is 20 people. Each color is a different person. The blue person here has a lot of this one, IL-7. The light brown person is IP-10. Orange person, MIP-1B, and so on and so forth. We all have different baseline cytokine levels and other components as well. And we think this poisons us differently for, because cytokines, you probably know, they're important for regulating our immune system. We think this poisons us differently for different pathogens. And I'll skip over this, but we all have different microbiomes. And I can tell you, if you gain weight or lose it, or if you get a viral infection, your microbiome will shift, but you will still look more like you than the person sitting next to you. So and that's true for other ohms as well. In general, these omic profiles, at least when fasted, they're fairly stable. So we have very strong personal profiles. And again, they'll shift some when you get one of these perturbations, but they're still very strongly personally imprinted. Okay, I want to spend the last part then talking about both our wearable work, and I lost track of when we started. When would you like me to finish?
You have at least 15 or 20 minutes.
Okay, so I'm going to tell you some of our wearable work, which is a lot of fun, and then I'll tell you the Exposome work, which is why Gary invited me here in the first place, I guess. All right. So let's start with the wearables. So you probably know there's this revolution going on, and just out of curiosity, how many of you are wearing some sort of a smartwatch or something like that? Only a few when they're in the back. All right, well, it's more, and a lot of you are just shy, I see. Okay. Well, it turns out at the time we started this about a little over 4 years ago, there were over 1,000 of these devices on the market, over 500 health-related ones. And so we went out and sampled about 30 of them that were accessible, and we thought, reasonable accuracy, we could get data, that's a big deal. And in the end, we settled on a number of them. Our cohort each wears a smartwatch and uses a Moves app, which tracks GPS and activity and such. That's a free app, you can download that yourself. I have 8 of these devices right now. I have 3 smartwatches. I normally wear a smart ring. I have this device I'll talk about a little more in a minute. Radiation exposure, they measure all kinds of things. You may not realize this. Looks like your radiation exposure is over 3 times that of Palo Alto. Okay, it's roughly the equivalent of about, I'd say, 27,000 feet. So don't worry, I don't think it's that. Well, I don't know how high that means. Okay. You see, old buildings will have that kind of thing too, it turns out. So anyway, as you can tell, they measure all kinds of things. They'll measure activity, heart rate, skin temperature, sleep, blood oxygen, that's pulse ox we use, so on and so forth. So you can measure many, many different things. Nearly all these things collect data and relay that into your smartphone. Okay, so the smartphone's collecting a lot of this information. The name of the game is to collect this information and figure out people's personal baselines and then look for deviations from those baselines under interesting conditions, like health or other things, other circumstances. So to give you a feel for this data, this is my data, a few months of data. This is my circadian patterns. This is sleep, heart rate, skin temperature. So I sleep at night, wake during the day, occasional nap, back to sleep at night, okay? This is my heart rate, low at night, high during the day, back down. Skin temperature, it's different from core temperature. High at night, down during the day, back up at night. That's my data. This is 43 other people's data. Everyone has a different baseline resting heart rate, blood oxygen, skin temperature, what have you. We're all different in our baseline measurements. This is activity patterns, so you can follow people's activities. These are people who are monotonic during the day, so not active at night. Generally kind of uniform activity during the day, back down at night. These are morning people, they spike in the morning. These are morning and afternoon people, morning, afternoon, evening. So you can group people. You can do this sometimes we do it visually. This is actually done using algorithms that can basically cluster people according to their patterns. And these are the 4 major patterns that did pop up. All right, so once again, you can define people's personal baseline, then look for deviations from that baseline. So this turns out to be one of my biggest deviations. This is blood oxygen. It turns out it drops on airplanes. This is known already, although I would argue it's not very well known. Even most MDs don't know this. But it turns out, so to walk you through this is a short flight, San Francisco, San Diego. That's altitude in green. Plane goes up, bubbles off, comes back down. That's my blood oxygen. Starts high, drops low, hits uniform cabin pressure, comes back up when it lands. What's not so well documented is how low does it go and how much does it stay low. So it turns out, we now know is an underestimate. This integrates all flights, but 70% of the flight or more, it'll be 96 or below. Remember that number, 96? These days, I can tell you, when it hits 30,000 feet, for me, it's usually 93 or 94. Person I measured coming over next to me on the plane here, actually, she dropped down to 88, 89. That's her thing, and that flight bounced around is between about 31 and 37,000 feet. She was always in that range. Okay, so that's, and for me, it's about 5% of the time I'm in that range. So, you know, that's not perilous, but it's, you know, not great either. So it turns out, with one exception, everybody drops. Doesn't matter what ethnic group you belong to, your blood oxygen will drop if that flight goes up high enough. So there's only one exception. Okay, so why is this significant? Well, this I couldn't find so well documented either. Well, it turns out it correlates with fatigue. Again, in hindsight, this seems obvious. You can do a blinded test, see whether you're tired or alert, score things. For me, it doesn't matter whether I'm on the ground here or in the air. Blue means they scored me as tired. Red is alert. And you'll notice that threshold is roughly 96. Okay. You can also do a reaction time test. This is blood oxygen, reaction time. So reaction times are faster when the blood oxygen's up, drop when it's lower. So the point out of all this is that when the plane goes up, your blood oxygen goes down, you get tired. So we think the reason you're tired on flights isn't because you've been working on grants too hard or partying too late or watching that Alabama game or Georgia game, whatever. It's that the oxygen's the pressure's low in the cabin, your oxygen drops, and you get tired. So if you're workaholic, that's disappointing news, like me, because you can go in all fired up and you'll probably still get tired. But it turns out, I say I left the slide out, but it turns out for long flights you can adapt. And for me, that number is 7 hours. So after 7 hours, I'll come back up. My oxygen will go back to normal. So on these long flights, you can actually recover. So that leads to the question, what about airline pilots and flight attendants? And so the one exception we noticed was a flight attendant sitting next to me who actually wore one of these things, and sure enough, she did not drop, actually. So she was adapted, we believe. Okay. Interesting, I tried a pilot, he did drop, so, okay, go figure. I know that sounds scary, but all right. All right, so again, these are they're amusing, they're very valuable, they're kind of nice to see. Here's a health-related story. which is, it turns out these devices were actually very powerful for me for first alerting to me when I got Lyme disease. So the backstory is I was in rural Massachusetts for for one day actually, helping my brother put up fences, and then 2 weeks later I was flying to Norway through Frankfurt. And on these short flights, your blood oxygen drops. And this is my normal drop because I measure myself all the time, hundreds of flights now. And this is on this particular flight where it dropped and the median was actually 90, which for a short low altitude flight is very unusual. And when I landed, it didn't come back to normal. It stayed fairly low. And I also noticed my heart rate was elevated, and I later learned my skin temperature was up. So we went and looked at that, and sure enough, this is one way of plotting. This is daily number of outlying measurements, true for median measurements as well. But it turns out that, so after discovering that, I then got a low-grade fever. I went to a doctor there in Norway who took blood, and my monocytes were up, and he said, yes, you have a bacterial infection. I warned him it might be Lyme. But he said, I think you should take penicillin. And I said, no, I think I should take doxycycline. As you might imagine, that was a little tense for a while there. He did give in, gave me doxycycline, and then it cleared it up. That's the day I got it here. And then when I got back, I gave blood, and sure enough, I was Lyme positive. And I had the perfect control because I'd given blood a few days before I left, and I was negative. So I seroconverted during that time. So the point out of all this is that you could actually tell from the drop in oxygen, from the elevated heart rate, and later, as with skin temperature here, that also drops. So that then alerted us to let's look at the 2 years of data I had at the time, find all the times when I had outlying heart rate and skin temperatures. And it turns out there were 4 of those periods. Here are the Lyme disease days. You can't miss those. Those are really, really strong signals. Turns out there were 3 other times, 2 of which I reported having a viral infection. One is here, and this one we now know is here. That was a viral infection as well. There was another time when I didn't realize I was sick, but it turns out I had a high heart rate, and then we looked at the data. I had high CRP, which is an indicator that you're sick as well. So every time I've had high CRP and have been sick, actually my heart rate was high and my skin temperature was low. So that tells you can pick this up. So then we wrote algorithms to be able to find from a personal baseline, a resting baseline. We now do it for times when you're not resting. When your heart rate goes abnormally high and alerts you, that's the goal. In this case, this is retrospective. So we looked at when there's a delta plot. It's a little complicated. But the point is we can see someone's heart rate go up before they report being symptomatic, including my own. Okay. So this is me, my 4 times of illness. We had 3 other people got sick, 1 twice. Every single time, we can see their heart rate go above baseline. And the reason is because these things take tons of measurements, 250,000 measurements a day. And so all you have to do, even if things are a little screwy, you get so many measurements you can't miss. In this case, it was set up with a 2-hour window. We now think we can do it for a half-hour window, where you can tell where people's heart rate is above background. Skin temperature doesn't work as well, but heart rate does. Okay, so the last thing I'll tell you is that we also think we can see differences between insulin-resistant and insulin-sensitive folks based on heart rate as well. So we think these smartwatches could be early alerters for other things as well. I forgot to say, so back here we're writing an algorithm and setting up a study. We're waiting for final approval, but basically the idea is to tell you when you're getting sick before you realize it. And that has, we think, lots of utility. All right, so maybe 5 minutes on the exposure. How's that? Okay. All right. So I mentioned all these other things about wearables and things all impacting your health. What about the exposome? So a little over 4 years ago, something just happened. Oh, okay. We started exploring the idea. We knew this was a big hole in that picture I showed you. We can now collect food, which we do, and other things. But the one thing we weren't collecting so well was our environmental exposures. So we set up a system to try to look at least aerosol exposures, both biologicals and abiotics, chemicals that are out there. So we took this device from, okay, it's acting up. Allison, I hope this doesn't, there where. We took this device we got from, it's a pen meter from RTI, thanks, sorry, mental block there. Anyway, we've actually modified in a way where we have filters on. It sucks in roughly 1/8 of what your breathing does, and it basically captures all biologicals, at least all bacteria. It's partially catching viruses, we think. So submicron stuff's tougher, but it's capturing it on a filter. We then elute things off that filter and sequence the bejabbers out of it for both DNA and RNA. And then we also have another filter in here where the air is sucking through with a chemical absorbent. So basically we're capturing biologicals and chemicals in two different traps, if you will, on this filter. I keep it near me at all times. And we've now followed 3 people with a fairly long period of time. That one's me, a little over 2 years. This is a person over a year. This is a person for about 3 months. And then we have other sporadic times we followed folks as well in different circumstances and things. I won't be able to tell you all this. One nice thing about following me and the other 2 folk, but me especially, is I travel all over the place. So we've actually sampled locations from all around the globe during this period. So basically, as I say, we'll take these things we capture on filters and we sequence very, very deeply, both for DNA and RNA. And I can tell you in aggregate, we collect over 2,500 species that people are exposed to of all sorts. I'll break this down a little more in a minute. But basically plants, bacteria, fungi, etc. On me, I've been exposed to roughly over 2,000 species we've identified. But even the person who only wore 3 months has something like 1,200 different species that he's been exposed to. Okay, so the point is you're getting exposed to a large diversity out there. So what does this look like? Well, it turns out this is following people over time. This is my profile. This is the other 2 folks. We change filters. We usually use them for a few days and then swap out devices. And this is grouped by fungi as this kind of yellowish orange. Bacteria is red. Plants are green. Even some animal things are blue. And you can see these profiles are very dynamic. So one period you might be very heavily fungal exposed, another period very bacterial, and so on. So that's actually just putting them by time. This is an aggregate of all samples across all time. This is actually clustering by days, if you will. So these are my plant days where I see lots of plant exposures. These are actually my fungal days, these these orangish ones here. These are heavy bacteria days and so on and so forth. You'll get exposed to different things at different times. Every time I'm in Monterey, this is one example. These 3 here, those are heavy fungal days. Okay, so where I stay in Monterey gives lots of fungal exposures, so on and so forth. So that's DNA. Here's what RNA looks like. It's mostly bacterial. So most of DNA we think is in things like pollen and such. It's not that active, so you're getting mostly DNA reads, whereas when you look at RNA, bacteria are growing and doing whatever they do, and so you get lots of active reads from RNA. We can then say, well, let's start clustering this and see where the impact is coming from, if you will. It's called the variance, for those of you who follow this sort of thing. This is all samples. These are my samples. Turns out the number 1 factor affecting the exposome is location. Okay, so when I'm in Asia, you get, that's more over here. That's these dots here. I have a hard time seeing this here, but you can break out different areas by location. This is the group as a whole. This is me in particular. But you can also actually tease out seasonal effects. We can find the particular organisms that are driving each of these. Which ones are driving, for example, your Asian exposures? Which ones drive your East Coast exposures? Which one and it's not all plants and things. It's fungi and other things. It's different species are doing this, and I didn't show you that, but happy to show you later. We can also do, even within a fairly defined location like the Bay Area, we had 4 people wear the device exactly the same time, 4 different devices, and collect samples from that. And even though they're in the same general area, everybody has their own personal exposure. And again, they cluster into people who travel more, like me. I'm more scattered in what I'm exposed to. And those who have who work and actually live in a very close environment, they have a very tightly clustered pattern. So, but it will be different. Every person's exposure is different to the biologicals. We don't have as much data, but we've collected chemical data for several months. on me and a few other folks, and we can see what chemicals people are exposed to. All right, somehow I lost the slide there. Sorry about that. Well, anyway, you'll be amazed. It's kind of an interesting slide. And I'm sure Doug will tell you about what his experience here, but you will see there are strong signatures with location with things like pesticides, carcinogens that you wouldn't necessarily expect. So it turns out DEET is all over the place. It shows up in almost all our profiles, but some areas are stronger than others. We can see some nice carcinogens coming up in certain locations that, could be a little bit scary. Again, sorry, somehow I lost that. But anyway, we can also then correlate chemicals with biologicals. So these are location, each of these is a period and a location. It's by time. And we can see which chemicals will correlate with which biologicals. And just to give you one fun example, in areas where there's a lot of, it's pyridine, it turns out there's low fungi. Okay, pyridine shows up in paint. And so you can start making these kinds of associations that in hindsight always make sense, but not always so obvious. So I'm in a house that was totally renovated when I moved in. They use all green stuff. I got a lot of fungi in my house, but if I were to walk into someone who used a lot of pyridine in their paint, they would probably be a low fungal house. Okay, so these are the kinds of correlations you can tease out and find. So net result out of all this is that we actually all have our own personal exposome clouds, and they're heavily influenced by what you're seeing. I have a cat, a dog, and a guinea pig. I can find signatures associated with each of those things in my exposome cloud, along with other, the usual plants and bacteria and other human things, tons of stuff living on us that are showing up in this exposome as well. So the bottom line, all of this is that data from all kinds of sources we think will impact your health, your exposures, your other forms of health. These are the people who did the work. Try and wrap up because I have feeling I'm over time. But the bottom line out of this is I've had some incredibly talented postdocs and students in my lab, Wen Yu Shao, Brian Pine, and Kevin Conchapontages, driving a lot of the omics profiling. The wearables was led by Shao, Jesse, and Dennis. And the exposome work is led by a very talented group of 5 folks. Chao Zhang, Xin Wang, Laura, and Ting have all contributed to that exposome project. And so if there's time, I'll take questions, but if I borrowed time, it's okay. I'll catch you later.
We have time for a couple quick questions, and we'll have more time at the end. So
Okay.
Questions, come to the microphone if you have a question.
Or you can shout and I'll repeat it, whatever works for you.
So I just want to hear your take on, you may have seen all these methylation papers coming.
Which one's the methylation? Is that what you said?
Yeah.
Yeah. Uh-huh.
They're using concepts like mendelian randomization to most of the complex trait associated methylation. Consequence of the disease rather than causing causal effects.
Yes, I know what you're getting at. Yeah, there's a lot of variants that will drive methylation, and that's what a lot of people see, and I think that's what you're referring to. So the issue is about methylation patterns and where are they coming from, and a lot of them are driven by actual genetic variants. So the genetics is driving changes in methylation. What's powerful about what we're doing, we're following the same person over time. My genetics has not changed. We've looked at that quite a bit, actually. So my genetics has not changed much as far as I can tell, but my methylation does. So I think we're controlled for that, and that's how we're dealing with that. Did I address your question properly? I think so. You're right. And I have those. I have a ton of that stuff, right? Right. And that's what we think we're following here. Yeah, when you follow someone over time.
So those changes, do you think, are they contributing to disease? Or once you have a disease?
Yeah, that's a great question. So the answer is we think yes. I didn't say, but I have this one will be more of a genetic variant. I have a variant that actually kills a particular gene, a phosphodiesterase gene, on my father's allele. My mother's allele is actually methylated in the promoter and makes very few RNAs. And the prediction is I should have higher than normal eosinophils, which I do. I'm right at the top edge of that. So I do think that these methylation patterns do influence health. Now, for my mother's, what I don't know, I'm guessing that one's actually driven genetically too, but by an unlinked, a mutation that's a little bit further away. So it doesn't exactly address the one you said. I'm assuming those times when my methylation pattern went off and my glucose changed, that was due to some external thing. We don't think it's cell population because, yes, that shifts for the first day or two, but then it goes right back to normal. So I think it's due to something I've always been intrigued by whether viral infection, for example, changes your DNA methylation pattern. Not really well studied, to say the least.
But you don't think changes in glucose causing those changes in methylation?
Well, in this case, because it preceded when I maximized my glucose, I think it's the other way around, but I don't know that for a fact, or they could possibly reinforce one another as well. I don't know the answer to that for sure. But we like the idea that the methylation might be preceding or at least part of the driver for this. I could see feedback loops feeding into this. That would be my guess.
So, Steve, can you get the mic there so we can hear you?
Yeah, Steve Chin from Biostat. Wonderful talk. So you certainly did a lot of omics experiment. I wonder whether all the experiments are done on whole blood?
Sorry, I missed part of it. So what about whole blood?
Is all the omics profiling experiment done on a...
Oh, yeah, I forgot to mention this. It's done on peripheral blood monocytes for the RNA and the epigenome. For the proteomics and metabolomics, that's out of plasma, although we have done it out of PBMCs as well for the proteomics. So, yeah.
So do you think it's possible to increasing this to other...
Yeah, it's a great question whether how much is whole blood a good surrogate for the rest of the body. My own guess is it's pretty good. We do a lot of work I didn't present with autism, and it's amazing how many signatures show up there that are neurological signatures. So I think a lot of these things that happen are systemic. A lot of people said, Mike, there's no way you're going to see any signature on insulin-resistant diabetes in blood. It's really a fat and liver disease. And sure enough, those signatures are actually stronger in the blood, especially the immune signatures, not surprisingly, than they are in the fat and the liver. So I think a lot of these things are systemic. Now, it's not going to capture everything, obviously, but I hope it captures a lot of the information. And if nothing else, it should be a downstream marker for issues that might arise. Yeah, great question, though.
So with all these wearables, how long does it take you to get through airport security?
Well, believe it or not, nobody ever pulls me over except for London Heathrow, where they always push my bag aside. They always do a mass spec swipe, and then they push me on my way. So, but otherwise, nobody really cares. They're kind of used to it, or not. I don't know if they're used to it, because well, some people I see quite a bit. But in general, yeah, it's never been an issue. And I usually throw them in the bin so they don't have to dig them out. But even if I forget, they don't usually flag it. So, yeah.
All right, well, let's thank Mike again and move on to Allison.